of myeloma. So I think this audience doesn't need to be reminded that there are multiple immune deficiencies affecting T cells, B cells, other cellular types in the vicinity of the myeloma cells. So tackling myeloma through immune therapy is something that we've been doing for a long time. In fact, the first immune therapy was probably thalidomide, and then now we have monoclonal antibodies. So these are the two modalities that we have today, and a lot of people talk about these in their talks on immunotherapy. I'm going to actually totally skip these. I'm going to talk about what we don't do today, which hopefully we'll be able to do in the future. Those are the CAR T cells, which we just heard a little bit about. Go a little more into detail on the data. Talk about the checkpoint inhibitors, a little bit about NK cells and vaccines at the end. We have time. So cellular therapy first for multiple myeloma. Obviously, the area that has got the most press is CAR T cells, which we're going to talk about. But then there's also data from one study on using TCR-based CAR T cells, TCR-based therapy directed at NY-ESO, and then another one looking at marrow-infiltrating lymphocytes, a take on the TILs from the old days of kidney cancer that when I used to look after solid tumors. So anyway, so CAR T cells, again, I think most in the audience are aware of what a CAR T cell is. So for sake of time, since we are running late, I'm not going to explain the construct. But what is the data? So the first data on the use of CAR T cells in myeloma came from Dr. Stetmeyer's group, and this was a case report in the New England Journal sent as a letter that looked at CD19 CAR T cells. This was obviously something that they had going for lymphoma at that time and ALL. So they did a small pilot of 10 patients with myeloma. They gave it after transplant. They had one patient who had a fairly long remission, and that was the subject of the report. The other 9 didn't do well. And 19 is probably not the best antigen for myeloma. Some people do feel that the myeloma stem cells may express CD19, but again, in itself is probably not the best target. So there are a number of other targets that are more, likely to work in myeloma. They are the classic CD138, CD38, CD56, kappa light chain targeting. And then there are newer targets that have come about in the last several years, of which BCMA is really the one that has got more the data at this point. So as far as BCMA-based CAR T cell therapy goes, there are 4 large uh, experiences out there, relatively large. When I say large, they're still short of 50 patients each. So there is the initial experience through the NCI, the Penn experience, the Bluebird experience, and the Legend Biotech experience, and we'll just go through these quickly. These differ in their vector, they differ in their co-stimulatory domain. So what we're going to I'm going to skip through all this. Okay, so first of all, the first report on CAR T cells with targeting BCMA came from the group at the NCI, and this was a late-breaking abstract at the ASH 2015 session. And here, the patients got CAR T cells after having failed multiple lines of therapy. There were 4 of 12 responses, and nearly all the responses occurred once you got to the higher dose of CAR T cells. Subsequently, nearly all these patients have relapsed from the NCI experience. They did a larger cohort at the maximally studied dose of 9 million CAR T cells, and they presented that at ASH last year, and this was 16 patients, and here they see that there were patients that had responses in the next slide here. So 81% overall response rate. So you need to give a therapeutic dose of CAR T cells. And then in this experience, they did see a fair amount of CRS. They did see neurological toxicity. The second experience is the experience looking at BCMA CAR T cells from Penn. So this is a trial that is still ongoing. They actually in contrast to most CAR T cell trials, did their first 10 patients without any conditioning, and they're going to subsequently use Cytoxan. They're not using any fludarabine even with subsequent cohorts. And what we see here again in a highly refractory group of patients, that they see efficacy. There are some MRD negativity also reported, but what we see in their experience is a fairly impressive rate of cytokine release syndrome, 89%, and then they did see neurological toxicity as well, and even a patient who had FRES. And this was 10 patients. But the larger experiences come from these two presentations. First is the Chinese Legend Biotech product that has now been, I think, brought into the United States in co-development with Janssen, and trials are starting. So this was at the ASH 2016 meeting that they presented on 35 patients. Their patients were probably a little less heavily pretreated than what we see in the United States CAR T cell trials. And also, we have no data on what these agents were. We know in China, there's no daratumumab, there's no carfilzomib for most part. Lenalidomide just has started getting some more traction. So these patients may have not had modern treatment regimens, but they did see a very impressive response rates of 100%, and there were some patients out more than 1 year, and can some of these patients be cured was the question. And certainly that was the promise with which CAR T cells have been introduced in multiple disease settings. There are cures, I think, to report in ALL and non-Hodgkin's lymphoma. In myeloma, as Maury said, I think we're too early to report whether there are going to be any cures. And the toxicity in this study was again actually pretty manageable in contrast to the experience from Penn. But as alluded to a few minutes ago, the most data comes from the Bluebird experience that was updated here at the ASCO meeting. The trial has had a phase 1 and a phase 2 dose expansion cohort. This has been presented at a couple of meetings already. What we see here is the composite and also data for the first time on the dose expansion cohort. When they did the dose escalation, they went up to as high as 800 million CD T cells, and they allowed for patients only if they had high BCMA expression defined as over 50%. In the dose expansion, they allowed patients irrespective of how much BCMA they expressed, and ended up with about half and half having 50% and half having less than 50%. And then they at this time said they're going to allow for the CAR T cells as long as they had at least 150 million CAR-Ts, and doses ranged up to 450. And this may be something that may be important in a minute. So what we see here is that the patients were highly pre-treated group of patients and had a median of 4 to 6 lines of prior treatment, had about 40% at high-risk cytogenetics, and had at least for the majority the modern drugs. In the escalation phase, about 48% had seen daratumumab. In the expansion phase, because it happened later, 86% had seen daratumumab. And what we see here is again the toxicity profile is probably a little bit better than what we've seen with the CD19 product. There is not much cytokine release syndrome that is grade 3 or higher. The neurotoxicity occurred in only one patient out of 43 that grade 3, that was a patient who had a prior history of a subdural hematoma, developed another subdural hematoma on the study. On this trial, the patients did achieve an ANC of over 1,000 by about 32 days, and 78%, and platelet count of 50,000 was achieved, and 55% by day 32. What they saw was comparable Cmax in patients who got doses of over 150 million cells. And they said there was durable persistence that is over 6 months in about 44% of their patients. And higher peak expansion was seen in patients who had a response. The tumor responses are shown here. If you look at the patients as a whole, the overall response rate, as Maury said, was pretty impressive at 95%. Those who got more than 150 million cells had a higher response rate than those who got less than those who got about 50 million. And then if you look at by BCMA expression, that was the expansion cohort, not the whole group, you see not much difference, but perhaps in terms of depth, there may be some difference if you had a high BCMA expression. But again, numbers are pretty small, and this is in the cohort that managed to get 450 million. And as alluded to, the median PFS of the whole population was 11.8 months. And for those who got at least 150 million, those who got less than that actually had a pretty dismal median PFS of 2.7 months. If you looked at patients who became MRD negative, the median progression-free survival was a little better at 17.7 months. So this obviously brings up, what is the price that we're willing to pay for this expensive therapy? At what point in the course should this be introduced? How can these things be made better? I think that some have said that you need to probably go for higher doses of CAR T cells to improve the results. Others have said that the responses seem to tailor off as the CAR T cells went away. So maybe you need to give multiple infusions, and certainly maybe you try it in a less heavily pretreated population. Um, so there is, really a lot of activity in CAR T cells ongoing in clinical trials. There are a lot of ways to improve upon CAR T cells experience with better constructs, with combinations, with earlier treatment. So this obviously is where the future will lie for these. As far as other cellular therapy experiences go, the experience from the group at John Hopkins by Ivan Borrello, published in Nature Communications a few years ago, looked at marrow-infiltrating lymphocytes. He took patients' bone marrow a few days before their transplant and put it in a cytokine soup, and then they got their transplant and they got their expanded marrow-infiltrating lymphocytes reinfused a few days after the transplant. And again, this was just a feasibility study to tell you that this was feasible. The overall response rate of 54%, it's impossible to know whether that was anything better than what could have been achieved by the transplant alone. A little more, I think, excitement perhaps with the NY-ESO TCAR cell, TCR T cells. As you know, these are cells that are not CAR T cells, but they also employ a viral transduction of the TCR receptor enriched to recognize NY-ESO. And this has to be done in people who carry a certain HLA-A2 haplotype, so it's not applicable to all myeloma patients. And so they saw in a highly refractory group of patients, for most part, who underwent a transplant, that these patients had 70% near CR rates. Once again, it was given in combination with transplant, so it's difficult to know what the transplant contribution was, but most of these patients were progressing or had a very poor response to their induction therapy, suggesting that there may be something to this. And this is now the subject of a larger multi-institution study. There's work going on with NK cells as well. There is this presentation by Nina Shah when she was at MD Anderson that looked at cord-derived NK cells using patients who were masked at least two HLA loci. And again, just proof of principle here. I put this up just to say that there are trials going with NK cells. There's a Celgene study looking at placental derived NK cells currently ongoing. So we'll have to see where that kind of cellular therapy pans out. As far as the other area of immune therapy that people are continuing to hope for, it is the checkpoint blockade. As far as checkpoint inhibitors go, both pembro and nivo as a single agent did not have any activity in myeloma, but trials looking at it in combination with IMiDs initially suggested that there could be potential for this. So this trial that was presented by and subsequently published by Dr. Samaguel from Spain looked at patients who had highly refractory disease and saw a 76% response rate when pembro was combined with len-dex. And even in patients who had had len-dex, there was responses seen. And then there was another study published with pembro-pom-dex in a POM-exposed population showing an 89% benefit. We had a publication from, again, the group at University of Maryland that looked at POM in combination with pembro-dex, showing a response rate of 60%. As you know, POM-dex usually has a response rate expected of about 30%. So these trials really increased enthusiasm for this kind of combination to be studied in myeloma. So there were multiple studies that were launched looking at a variety of checkpoint inhibitors with IMiDs in myeloma. The two studies shown here were the ones that got off the ground first: pembro-pom-dex versus pom-dex in refractory patient, and pembro-len-dex versus len-dex in treatment-naive patients. And we saw alert from the FDA about a year ago now that actually showed that this combination may require a little more caution, and they put on hold not only these two trials, but every trial combining IMiDs with the class of checkpoint inhibitors. And so for the first time at the ASCO meeting, data was presented by the FDA. So it was kind of maybe one of the first times the FDA presented data for the first time for their trials that they put on hold. And they show why they did so. So this is the relapse refractory trial, pembro-pom-dex versus pom-dex. The hazard ratio was 1.61, suggesting that there was an increased risk of death when you gave the drugs together, combined compared to pom-dex alone. The median follow-up, which has been criticized, is just 8.1 months. And they looked at serious adverse events and saw that there were higher rates of serious adverse events in the pembro-pom-dex arm. And then in the upfront study, similar results were seen, an even higher risk of death when you gave the drugs together. Again, higher rates of SAEs. So they went ahead and did an analysis to say what was the role of these side effects in terms of even predicting responses. So immune-related adverse events was a term that they coined, and I didn't put up the definition there, but they had like 50 things that they coined as immune-related adverse events. And as we'll see in a minute, there may be some reason why you could fault that. But first of all, in this analysis shown on this slide, they tried to see if immune-related adverse events, which were grade 3 or higher, led to better responses, that if maybe the toxicity was higher but the responses were better, you could perhaps justify it. And they showed that in the relapse refractory trial, the 3-drug arm didn't seem to have any greater efficacy if you had greater immune-related adverse events. When you look at the upfront study of pembro-len-dex, it seemed like, yes, when you got side effects, you had better chances of response, not only in pembro-len-dex, but also in the len-dex arms. But what has caught people's attention is how high the rates of immune-related adverse events are reported with pom-dex and len-dex. So nearly 45% and 44% of patients were said to have had immune-related adverse events with these drugs, and grade 3 or higher, those smaller numbers, 13% and 8%. And we don't usually think of that high a rate of immune-related adverse events when we use pom-dex and len-dex. So I think that this trial is still something that the field is trying to come to grips with, and Dr. Badrus um, was a discussant on the presentation that looked at the efficacy of this. This was from a poster discussion at the same meeting, and his observations, because he did the initial phase 2 study, are worth pointing out, because in his experience he hadn't seen this kind of toxicity. The median age, he said, was older. Perhaps that played a role. There were, in the Pembro arm, more people with renal insufficiency, more high-risk patients. So that is something that may have played a role. Also, where the side effects were coming from has also been a thing that people are talking about. Where did these occur? Were these in places where they didn't know how to give Pembro? So I think that this is something that uh, is still open to interpretation, because the follow-up at the time of reporting and stopping the trials was just about 6 months. And what if you had gone longer? Because with immune therapies, it's thought that the benefits accrue later. Could you have had a different result? Uh, However, the thing is that as soon as this presentation was over, the hold on all other IMiD checkpoint inhibitors was lifted by the FDA. So trials are restarting with other checkpoint inhibitors and IMiDs. So because in those trials, at least, the FDA didn't find any evidence of increased toxicity or death. And then the last slide, just to say that there are moves afoot, obviously, to continue to develop vaccines. This is a large CTN study looking at three arms: Len alone, Len with GM-CSF, and Len with GM-CSF, and a MAGE vaccine that is currently accruing. So to conclude, IMiDs and monoclonal antibodies are currently available immunotherapeutic options for myeloma. Multiple cellular therapy options. CAR T cells are hot still. The marrow-infiltrating lymphocytes, the TCR T cells, NK cells are being explored. We have hit some road bumps with checkpoint inhibitors and IMiDs, but the trials have restarted, and trials with vaccines are also ongoing. So that's all I had to say. Thank you.